Chapter 15, Microbial Mechanisms of Pathogenicity. In this chapter, we're going to look at how microbes enter the host and adhere and then actually cause disease. So one of the prerequisites for causing infection, obviously being able to gain access and being able to hold on to then cause the disease. When we talk about pathogenicity is technically the ability to cause disease. Virulence is the extent of pathogenicity, a measurement of the severity of the disease produced. People always interchange these two terms. I must say, I do that myself sometimes. However, they are technically different. When we talk about the portals of entry, there's three basic portals: the mucous membranes, skin, and the parenteral route. Mucous membranes are the most common. Of the mucous membranes, respiratory is the most common site for the portal of entry. Skin, things get in through hair follicles and pores and sweat ducts. The parenteral route is what happens when you have a needle stick or something that allows the deposition of the organism beneath the skin or membranes. Each organism has a preferred portal of entry, and that's pretty much the prerequisite for them being able to cause the disease. For example, Salmonella typhi is the causative agent of typhoid fever, a nasty GI disease. We talked about that with Typhoid Mary, and it causes what is referred to as dysentery, a bloody diarrhea. The portal of entry is going to be through the GI mucosa, so being swallowed is its preferred route, and if that occurs, then you can get the disease. If you get the organism on your skin, maybe you get a slight inflammation, but you're not going to get typhoid fever. So that's why certain organisms cause respiratory diseases, others GI diseases, skin diseases. And you'll see when we start to talk about our microbial diseases, we're going to take an approach that is going to look at things systematically. So we will talk about GI versus respiratory versus cardiovascular, central nervous system diseases in the subsequent chapters. Besides the fact that the portal of entry, there's also a number of invading microbes that is needed in order to be able to cause infection. So we can measure this by what is referred to as an ID50. So this can help us see the virulence of a microbe. The ID50 is the infectious dose for 50% of the test population to be infected. For toxins, we usually measure the lethal dose. Again, 50% of the test population would be killed by a particular toxin to measure its LD50. Looking at Bacillus anthracis, which is the causative agent of anthrax, provides an interesting comparison in looking at the different types of the disease based on their portal of entry. So when you look to the right, you see a skin lesion from what is referred to as cutaneous anthrax. The ID50 to get the cutaneous form is only about 10 to 50 endospores. The inhalation form is a more serious disease, but takes about 10,000 to 20,000 endospores in order to cause the inhalation form. And the ingestion form, it needs about 250,000 up to 1 million endospores, where you have to have that high of a dose in order to get the ingestible form. That form is actually almost 100% lethal. So if you look at what happened with the anthrax in the postal facility when we had the case of bioterrorism in the United States, most of the cases were the cutaneous form because of the lower threshold. It had an ID50 of only 10 to 50 endospores, and so it was much easier to be able to cause the skin variety. If someone happened to get a very high dose, then there were individuals that did get the inhalation form, and we did see an occasional ingested form as well. For the toxins, here are some of the toxins that you might be familiar with. The botulinum toxin from Clostridium botulinum, causative agent of botulism, is one of the most deadly toxins that we know. It's estimated that the amount of toxin on a period on a paper can kill 30 people. So if we calculate that, it actually has an LD50 of 0.03 nanograms per kilogram. Shiga toxin from Shigella, which causes a nasty GI disease, it's kind of moderate, 250 nanograms per kilogram. And the staphylococcal enterotoxin that causes staphylococcal food poisoning takes a much larger dose, 1,350 nanograms per kilogram, in order to cause the disease. So not only does the organism have to get in, it has to be able to hold on. Many organisms have what are referred to as adhesions or ligands that actually bind to receptors on the host cells. We talked about this a little bit in chapter 4. So we talked about things like the glycocalyx or the capsule that's found on things like Streptococcus mutans to be able to allow it to hold on to teeth and cause biofilms. Streptococcus pneumoniae, Klebsiella pneumoniae, these are causative agents of various forms of pneumonia, and they use their capsule to be able to adhere. The respiratory system, your ciliary escalator, and all that mucus is constantly trying to move things up and out, and so therefore it's important for the organisms to be able to hold on. For strep pneumo, if you do not have the capsule, the bacteria is not virulent. We see the same principle of adherence with biofilms. Remember, we think about 70% of all bacterial infections probably occur as biofilms. The microbes start to make an extracellular matrix, which is basically this gelatin-like substance that really protects the microbial community and also, like Jell-O, helps it to adhere. So holding on and then providing this protective hydrogel means the biofilms are not only able to proliferate, but also resistant to disinfectants and antibiotics. Other examples of adherence, we talked about the fimbriae in chapter 4. So those needle-like projections on things like E. coli that allow them to stick on. M proteins, same principle, but just specific proteins for Streptococcus pyogenes, OPA proteins for Neisseria gonorrhoeae, and even the spirochete, the Treponema pallidum, those tapered ends help them to adhere as well. When we talk about this adherence, though, it's not just simply a Velcro ball sticking up against the wall. It's more of a direct interaction. So here we see those adhesions or ligands, the green projections from the pathogen, and how they interact with their complementary surface receptors. You cannot see that when you're just looking at the pictures. In the bottom, we see E. coli in the urinary bladder and bacteria on the skin. However, the receptors are present, allowing things to adhere to be able to hold on. So besides being able to hold on, we also see that we're going to have pathogens that can do damage to the host cells. We'll talk about some of the direct ways that cells can be damaged by bacterial entities. So direct damage can be from things like enzymes to disrupt the host cell, things like hemolycins that we talked about with red blood cells, but also the waste products from various organisms. By far, most of the damage is going to be done by toxins. If we look at various virulence factors, we see that a number of pathogens are going to be able to secrete enzymes that allow them to avoid being killed and also to invade tissues. So exoenzymes such as coagulase allows bacteria to be protected in a fibrin clot. So with coagulase, it binds to the surface of the bacteria, an example being Staph aureus, where it coats the surface when it comes in contact with the blood. That fibrin clot then forms and it protects the bacterium from phagocytosis. It also allows the bacteria to proliferate within that clot, and therefore it allows the bacteria to be more virulent. Then these same organisms often use kinases that then digest that fibrin clot. So this then allows the bacteria to escape into circulation and to infect other tissues. So we see in the diagram here an example of the coagulase and a specific kinase, streptokinase, that's allowing the organism to get into circulation. Other exoenzymes may directly work on the tissues themselves. So things like hyaluronidase is going to basically interfere or digest the connections, kind of the tissue cement, in between cells, and the collagenase digests collagen. Both of these then allow the bacteria to be more invasive and get deeper into tissues. Some pathogens also make things like IgA proteases. These proteases specifically destroy a certain type of antibody, what's referred to as immunoglobulin A. This is the main antibody that is found at all mucosal surfaces. So things like Neisseria, Haemophilus, and Streptococcus, they make these IgA proteases that digest the antibody so it's no longer effective. We also see virulence factors from some bacteria that are known as siderophores. Remember that bacteria need iron to be able to grow. So some bacteria can secrete these siderophores that bind to iron. Iron most of the time is not free. It's usually bound in the blood and to things like transferrins and lactoferrins and such. So what happens is that these siderophores bind the iron, and then the bacteria has bacterial receptors for the siderophores. Once it then gets that iron, then it is able to actually rapidly divide. We also see other interesting strategies for cells to be able to gain entrance. So things like Salmonella and E. coli, they produce what are referred to as invasins. These proteins are very unusual in that they actually rearrange the actin filaments, which is the cytoskeleton of your cells. So they allow the entry of these pathogens for the initial stage of infection. You can actually see them almost puddling into the cells, and I've included a little HHMI video that looks at how a salmonella actually causes infection, and you can actually see them kind of being swallowed up in the process. By far, most damage is going to be done by toxins produced by bacteria. So we see the ability to produce a toxin is quite important for many of our very prevalent pathogens. So in order to protect us against diseases where the toxin is the problem, we often have vaccines that are made against a toxoid. So that's an inactive form of the toxin that can be used for a vaccine. For treatment, however, if somebody has this toxin, we may use an antitoxin. This is specifically going to be antibodies produced in someone else or something else so that we can give the patient preformed antibodies against that specific toxin. An example of this would be tetanus. Clostridium tetani, remember, is the gram-positive rod that forms endospores. And so when someone gets tetanus, what happens is that they usually get a deep puncture wound, and then the endospores are able to enter that wound. Those types of wounds usually heal from the outside first, forming a nice anaerobic environment, and then the endospores germinate within that deep puncture wound. They then start to produce a nasty exotoxin. This tetanospasmin causes everything to go into a tetanic spasm. The muscles can no longer relax. So you see the outcome of the opisthotonus, where the muscles have contracted so hard that you can actually see the arching of their back. Unfortunately, this is a terrible death, and that is why we use the toxoid form to vaccinate people in our DTP vaccine. The treatment, however, one of the first signs of tetanus is lockjaw, and if you want to save your patient, you need to be able to give them these antitoxin antibodies to bind up the toxin. So we see a good example here of how we use toxoid to vaccinate and the antitoxin antibodies to be able to treat when people start to show signs of tetanus. So now we're going to compare the mechanisms of exotoxins and endotoxins. The tetanus exotoxin was an example of these exotoxins that exo means that they're secreted. So these are most commonly made by gram-positive bacteria, and it's part of their growth and their metabolism that they secrete these exotoxins. Endotoxins, on the other hand, are part of the LPS, the lipopolysaccharide layer of gram-negatives. Specifically, the lipid A is what's said to be toxic. So these endotoxins are liberated when the bacteria are dying and their cell walls start to break apart. So if we look at exotoxins, they are mostly made by gram-positives, though we'll see a few exceptions when we talk about specific diseases later in the term. They're a metabolic byproduct that is produced by growing cells, and their chemistry, they are proteins. Proteins are antigenic, meaning that they're good at stimulating antibody production. That's why we can use antitoxins, antibodies that have been produced in something else, preformed, to neutralize the toxin. These do not cause fever, but they are very lethal. So their LD50 is very small. It only takes a very small amount in order to do harm. So now we're going to look at some of the subcategories, the different mechanisms of actions of various exotoxins. So type 3 exotoxins are what are known as AB toxins. They specifically inhibit protein synthesis. They have a portion and a B portion. The B portion is the binding portion that is going to interact with the receptor on the host cell. If you can prevent the interaction of the binding portion and its receptor, then in theory you can prevent the pathogen from getting in. However, once the pathogen gets in, it then rearranges and the active form is then released and it inhibits protein synthesis. If a cell cannot make proteins, it obviously is going to die. One notable example, Corynebacterium diphtheriae, the causative agent of diphtheria, makes a nasty toxin that forms a pseudomembrane in the throat and literally suffocates the patient. Other exotoxins, type 2 toxins, are membrane-disrupting toxins. We talked about these when we talked about the hemolysins on our blood agar. So in that case, we were using the hemolysins to help us differentiate some of our pathogens. But think about that if these types of toxins are being employed in the body, rupturing red blood cells is problematic. And also, this is one of the ways that the bacteria tries to get iron. There's also leukocidins that kill white blood cells and some other type of membrane-disrupting toxins that can kill other cells within the body as well. The type 1 toxins, or superantigens, are kind of the most bizarre of the exotoxins. These cause a very intense immune response where it causes the immune system to release a whole lot of inflammatory cytokines. You get what's referred to as a cytokine storm. It causes fever, nausea, vomiting, and diarrhea very quickly. But the real problem is that these inflammatory cytokines are causing your vessels to become leaky, and you start to have a drop in blood pressure. People often go into shock and can die from this condition. One of the notable pathogens, Staph aureus, and its association with toxic shock syndrome is because of these superantigens. So if we look at the exotoxins, they are specific for a certain structure or function in the host cell. So when naming these, we see things like the toxin from Clostridium botulinum and Clostridium tetani. Those are referred to as neurotoxins because the Clostridium tetani causes everything to go into a spasm, so everything contracts, whereas Clostridium botulinum, a neurotoxin, causes flaccid paralysis. Other ones, we see the Vibrio cholerae and Staph aureus causing staphylococcal food poisoning. Those are referred to as enterotoxin because they are working on the GI system. We also see some interesting patterns here. If you look at the column for lysogenic conversion, we see some of these notable pathogens can only make these toxins when they have gotten the virulence factor, the toxin gene, from being infected by a bacteriophage. So we see that the only one in this list, Clostridium tetani, it makes its toxin all the time. However, the other ones only when they're infected by these bacteriophages and they incorporate that toxin gene into their genome. So one way to prevent these diseases would be to prevent them from acquiring that toxic property. Endotoxins aren't as lethal as exotoxins, but they're still problematic. So endo means in or part of. So when we talk about the endotoxins, that is the LPS layer of gram-negatives, specifically the lipid A portion. What happens is that a gram-negative bacterium comes into the host, macrophages do their job and ingest the bacteria. They then start to digest the bacterium. This is going to release those endotoxins, and it's going to cause the macrophage to produce cytokines, specifically things like interleukin-1 and tumor necrosis factor alpha. These then are going to go into the bloodstream, and one of the things that they can do is travel to the hypothalamus of the brain and start the production of prostaglandins. These reset the body's thermostat, and you end up running a fever. That, in essence, is a good thing. Fever is actually a good mechanism to help you fight infection. But these endotoxins can also start to do damage when the TNF-alpha and other inflammatory cytokines are produced. They can start to cause what is referred to as endotoxic shock. So when that gram-negative bacteria is ingested by the macrophage and things like TNF-alpha are then released, they start to damage blood vessels and cause them to become leaky. You then have loss of fluid and a critical drop in blood pressure that again could cause shock and be potentially lethal. When we refer to septic shock, that can be caused by any bacteria. Endotoxic shock is specifically from gram-negatives. It is important that we look for these endotoxins. They actually are very heat stable, and so we want to make sure that we're testing for these endotoxins in various solutions that could be potential injectable or infusible products. We actually do a really weird assay, what is referred to as the LAL assay, which stands for Limulus amoebocyte lysate assay, that tests for the presence of these endotoxins. It uses amoebocytes, which are white blood cells from horseshoe crabs, to see if these endotoxins can stimulate their clotting pathway. So this clotting cascade is going to be activated by endotoxins if present, and we have made this into an assay so that these clotting enzymes will cause a substrate to turn a color so we can easily detect when the endotoxins are present. We definitely need to know that because injecting these endotoxins could also cause that endotoxic shock even though no bacteria is present. This is actually still a widely used assay that uses horseshoe crabs that are caught from the wild. So here we see horseshoe crabs being bled to test for endotoxins. You might note that their blood is actually blue. And if you look, it's actually a sizable volume that is being removed. These are then released back into the wild. However, we are not sure how detrimental this is to the health of the horseshoe crabs. And because we need this assay, we actually are doing harm to the population. People are trying to look into different tests, but at the moment, the LAL is still the assay of choice for looking and testing for these endotoxins. So endotoxins, in summary, are made from gram-negative organisms because it is the LPS, that outer membrane. That lipid A is an unusual lipid in that it's very heat stable. So that's the problem. You could actually destroy the bacteria without destroying the endotoxin, and that's why these endotoxins can still be left in these various solutions. It actually also causes fever, as we saw in the previous diagram. However, these are not very good at stimulating antibody production. Lipids are very repetitive, and so they're just not very good at eliciting an antibody immune response. So therefore, they are not neutralized by antitoxins. We can't use antibodies to help with the situation. The only good thing, they have a relatively large LD50. That means that it takes a relatively large amount of the endotoxin to cause something like endotoxic shock. So in this example, we see wash water was containing Pseudomonas was sterilized and used to wash cardiac catheters. 3 patients then developed fever, chills, and hypotension following this cardiac catheterization. The water and the catheters were sterile. There were no bacteria present. So the question is, why did the patients show these reactions? It was from the endotoxins. So again, things can be sterile, no bacteria, but the endotoxin may still be present. So these should have been tested to make sure that they didn't have the endotoxins by doing the LAL assay. Bacteria are not the only pathogens. We looked at the eukaryotic pathogens in Chapter 12, so our parasites and protozoas and fungi, and viruses in Chapter 13. Now we're going to look at some of their pathogenic properties as well. So when we talk about viruses, they have to get into a cell in order to be able to cause damage. So we refer to that as cytopathic effect. We see some examples. Poliovirus actually causes a killing of the cells, so the cytocidal effect. We see things like the rabies virus has these acidophilic inclusion bodies in the cytoplasm of the nerve cells that are in the brain. So we see here Purkinje cells, and what are referred to as Negra bodies within the cell would be indicative of an animal that had been infected with rabies. This is often what had been done if a rabid or a suspected rabid animal bit someone. You would have to then, unfortunately, put the animal down and look for these cells upon autopsy in their brain tissue. Other things like cytomegalovirus is a very common virus that many of us probably have been exposed to, and this causes, at best, a mono-like disease. However, if we look at the cells, we can tell when they have been infected because we see these acidophilic inclusions in the nucleus and cytoplasm. It also causes the cytoplasm to swell up, hence the name cytomegalovirus. Things like measles cause cells to fuse, and other things like the polyomaviruses, they cause transformation. Remember, transformation in culture means that cells begin to lose their contact inhibition. They start to rapidly grow and basically grow over top of each other, much like they would a tumor in vivo, so in the host. And so we can say that we know when things are transformed when we start to see this uncontrolled proliferation. The fungi can also have some pathogenic properties. Some of this is just caused by their waste products, and fungi are notorious for causing an allergic response. They are very prolific in putting those reproductive spores out into the environment, and many people do have allergic reactions to them. However, they can also make things like the trichothiazine toxins inhibit protein synthesis, similar to what we saw for the bacterial toxins. They can make proteases that can do some cell and tissue damage. They can have capsules that prevent phagocytosis and help in adherence. The picture here is of Cryptococcus, a yeast, and you see the very large capsule around it. It actually causes a fungal form of meningitis. Other toxins may not necessarily be ones that are particularly damaging to cells, but things like ergotoxin causes a LSD-like toxin that is a hallucinogenic. Cleviceps is one of the causative agents, and you see in the picture what is referred to as ergo on rye. We always want to screen for these products as they come to market. Other toxins that you may have heard of, aflatoxins every once in a while are in the news and are responsible for recalls. So you may have seen a recall on peanut butter because there were aflatoxins. The problem with these toxins is that they are carcinogenic. So Aspergillus makes these aflatoxins, and it's a very common fungus out in the environment. You see it growing on corn here. So once again, we need to inspect our agricultural products to make sure that these fungi are not present. Other mycotoxins, things like the Amanita mushroom, makes a very potent neurotoxin. Fortunately, this mushroom has a bright red color, so it's kind of a definite warning, but not all mushrooms do. That can produce some interesting neurotoxins. Algae as well, we think of them as being good things in their photosynthetic property as primary producers. However, we also saw examples of the dinoflagellates being able to make neurotoxins. They specifically make what are referred to as saxitoxins that cause the paralytic shellfish poisoning. The ciguatera is the common name for that, and so we see here the example of the red tide that could potentially be toxic to humans if large amounts are ingested. The protozoa and the helminths, or worms, they're kind of more the typical parasite. They want to hang out in the host and really don't do much damage because they don't want to lose their host. The presence of protozoa can cause problems because of waste products. If you get a high load, then obviously they are producing a lot of waste that could interfere with host cell function. But they're also notorious for doing what is referred to as host defense evasion. So they get around the host defenses by one, some of them can grow in phagocytes so that they're just not killed by white blood cells that are trying to monitor the host system. And they also have antigenic variation. They change their look to the immune system. Some of the protozoa can, though, produce some proteases as well that could cause potential cell lysis. So things like Entamoeba histolytica, a nasty GI pathogen, can do some cell damage. Presence of helminths, the worms, the picture here, the Ascaris lambricoides is one of the most common of the worm parasites, being a nematode. They literally just want to hang out and reproduce in the host, living off their tissues and fluids. But they don't tend to make enzymes or any types of toxins, so most of the problem is going to be from parasitic load. When they get too high in number, they often then interfere with host function, and their metabolic waste products can also cause host function damage, as that may interfere with cellular function. So for some of our parasites, we see that their ability to cause disease is because they evade the host immune system. So antigenic variation occurs when the pathogen is able to change its surface protein coat. One example is Trypanosoma gambiense, the one that causes African sleeping sickness. This is the parasite that you would see as a wavy kind of worm with a flagella swimming around the red blood cells. The problem with the trypanosoma is that the immune system recognizes it, so it's always winning the battle but never the war. So what happens is that you have the first antigenic coat being recognized by the immune system. In this case, clone A has a pink coat, so the immune system will respond and clear that particular variety, but then a second one emerges with a new coat. Then that proliferates, and this keeps going on and on. So again, the immune system is going to recognize it and win those individual battles, but it never wins the war. And this becomes a chronic infection where these individuals literally just end up sleeping to death. They can no longer maintain a sleep-wake cycle because of the neurological damage. Other pathogens that you know, things like influenza and Neisseria gonorrhea, HIV, they employ similar strategies, which is why it's been very difficult to make vaccines for these types of pathogens. Another even more weirder case is measles. Not only is it fairly good at getting around the immune system, there is also an article that was put out last year in 2019 that said measles virus may wipe out immune protection for other diseases. they theorize that this is causing immune amnesia. So the measles virus increases the vulnerability to all kinds of other infections for months after people have had the measles, possibly even years after they've recovered. When the measles vaccine was made and introduced in the 1960s, the rate of childhood death actually fell precipitously, more than what you would have expected just from protection from measles as well. So they theorized that the vaccine was actually helping to protect from some of these other diseases. Because when you get the measles, apparently it is causing the antibodies to be lost. So they found when they looked at children that had been tested after having the measles, that on average they lost about half, if not as much as 73% of their overall antibody diversity. So what they theorize is that the measles vaccine seems to provide protection from other things more than just the measles, because the actual measles could damage the immune system, so the body loses its antibody memory to other antigens as well. And so that's why it's important not only to protect people from the measles itself, which is highly infectious, it's also protecting them probably from some other diseases that, again, even if they had antibody protection, that might be forgotten if they would get a case of the measles. So what goes in must come out. The portals of entry are similar to the portals of exit. So if something came in through the respiratory tract, we see that is probably the portal of exit. So things like coughing and sneezing. GI pathogens that come in are going to go out the same way via the feces or saliva, and things in the genitourinary tract as well. So urine and vaginal secretions are going to help provide exit for these various pathogens to be then passed on to another host. Skin, not only from contact, but also remember, we do shed our skin. Our skin does flake off, and hence the microbes with it. Blood is not accessible just outright, but when we have any broken skin or things like biting insects or needles and syringes, these are all things that can get under the skin, so that parenteral route to gain access to the blood. So in summary, we see the microbial mechanisms of pathogenicity is a balance between the host and microbe. When the microbe starts to tip things in its favor, that's when they begin to cause infection. So they need the right portal of entry. Again, the mucous membranes are always the most used portal of entry. Of the mucous membranes, specifically, the respiratory tract is the one that is most used. Skin is a portal of entry. The follicles are pores, sweat ducts, and the parenteral route, something that gets under the skin, so the proverbial needle stick. We also see that the number of invading microbes is important. So we look at the ID50. How many does it cause, or how many does it need to be able to cause infection? Even if you have a number of microbes, if they can't adhere, then the body is going to try to clear them. We constantly have things moving up and out, whether it be the respiratory tract or our GI tract, whatever, through the urinary tract. Things are flowing out in the form of urine. All these things are designed to try to prevent things from adhering. If they are able to hold on, adhesions and various things that allow them to hold on, then they have a better chance of penetrating and being able to cause the host problems, so virulence. So things like capsules and enzymes, those invasins. But by far, most of the virulence is from toxins. So damage to host cells from things like these toxins, both exo and endotoxins. Remember, some of this is occurring by lysogenic conversion for those bacteria. And we also see a variety of cytopathic effects. As we said, the portal of exit is similar to the portal of entry. So in generally, the same portal that was used for entry is the same one for exit. And we see now that microbe, that pathogen, can now be passed on to another host. As always, make sure you check your understanding as you go through the chapter.